Great, thank you so much, Yana. I'm Kenza. I'm going to be the session chair for this morning, and I'm very excited about the next couple of talks that we're having. So we're starting off today talking about some microfluidics and lab automation. So that's exciting, and then this talk is going to be given by Georgi Tanev, I believe. And the title of the talk is the Bioware Cyber-Fluidic Platform, a holistic approach to digital microfluidics. So Georgi, you can take it away.
Thank you, Kenza. Try to share the screen. All right, let me know if there are any technical challenges on the way. Good. Let me just shuffle things around. Welcome all, and thank you for tuning in for my presentation. My name is Georgi Tanev, and I'll talk about our cyber-fluidic system and why we believe that a holistic approach towards digital microfluidic is the way to go for establishing full cyber-fluidic systems. Let's start with looking how wet lab environment looked 50 years ago and how it looks today. Well, it's what we see on these pictures is that the lab environment pretty much remains the same. We still store all the chemicals in vials and different bottles on shelves, and we do perform reactions by just moving them from one tube to another. Well, yeah, we have to admit that a lot of new lab automation has appeared in terms of robots and modern instruments, but the underlying principle remains the same. You move fluids from one container to another through tubes or through pipettes. 50 years ago, also the field of microfluidics emerged, and in the 1990s, the microfluidics start actually promising that they will disrupt the wet lab field by providing unpaired automation capabilities in terms of scaling down the reagent volumes as well as automating all the handling. That was also marked with the invention of the application of PDMS for fabricating continuous flow microfluidics, and the next 10 years we saw quite a few advancements in that domain. What's interesting for us actually is happened around year 2000 when the digital microfluidics were first introduced to the research field. And it's interesting because it provides a completely different approach to handling fluids. Instead of having the fluids sitting into different tubes or bottles and moving them around with pipettes, we have droplets which represent both the container, the fluid container, as well as reaction chamber. The digital microfluidics offer a mechanism where if you have a droplet sitting on an array of patterned electrodes, by applying high voltage on neighboring electrodes beneath the droplet, you can make the droplet move, which means you can set two droplets on collision paths so they will merge or split them, dispense them from reservoir, dispose them, and apply some other controls like cool them up, cool them down, or heat them up, as well as implement different sensing techniques on these chips. That allows the creation of cyber-fluidic systems where this very elegant actually control mechanism allows the cyber-fluidic systems where we have a digital biochip to be paired with a computer system, ultimately providing reagent storage, reagent manipulation, and also other forms of processing, essentially allowing to implement full lab scale, full benchtop scale protocols on these small devices. Well, that inspired our cyber-fluidic platform, which we call Bioware CFP. And since the field is cross-disciplinary, we have actually, and the system becomes quite complex, we have split the system into three main parts, which are namely the fluidic system, the instrumentation system, and the virtual system, where, as the name implies, the fluidic system handles all fluidic operations, meaning moving the droplets or interfacing them with different sensors, actuators. The instrumentation system takes care of instrumenting the digital biochip, meaning providing all controls or resources to the biochip. And then the virtual system is actually the controller which orchestrates the fluidic operations. So the fluidic system consists of a digital biochip, which is the heart of the system. In our case, we based our design on a standard printed circuit board with a large array of individually addressable electrodes. We also have reservoirs on the sides, and a notable thing is that between each two columns of electrodes, we have individual heaters, so we can create different heating regions on the chip. We'll get to that later again. The instrumentation system, it's a hierarchical embedded system, which consists of the base control system, the brain of the instrumentation system, and a modular instrumentation subsystem, which is made in a way that the instrumentation system itself can be changed and adapt to the instrumentation needs of the digital biochip. To put it into perspective, this is a sampled version of the minimal system configuration that we have. The center is again the digital biochip, and on the sides you see two electrode driver boards, high voltage power supply, and the base control system. What this system allows us to do is we can actually programmatically move droplets from the external controller, which is the virtual system. And what happens is that the computer sends actuation sequences, commands, down to the digital biochip, and it makes these droplets move into the pre-programmed patterns. So as I already mentioned, the obvious and the straightforward approach is if you want to move a droplet from location A to location B on the chip, what you need to do is you simply need to write an actuation sequence, meaning you need to activate the electrodes one by another on the path which you want the droplet to take. And that's feasible and pretty straightforward for small applications and small chips. But when things start growing up and the chips become larger, in this case 640 electrodes, scripting starts being difficult. Another problem with the direct scripting is that it doesn't provide flow control, so you cannot actively monitor what's going on the chip and take different droplet paths or change your experiment on the go while it's running. So we start looking into what's actually needed in terms of tool chain to be able to get an application and to execute on the platform abstraction in an easy and rather simplistic way. An example for where we can start, how we can start implementing this system is, for instance, when we have to mix two fluids, A and B, we can tell the computer that these fluids are somewhere on the chip, and we can let it figure out a path to where these two droplets will actually merge, and that would create our another droplet, let's call it C. So C actually equals to droplet A plus droplet B. And then the mixing again is just moving the droplet in circle around on the chip until gets homogeneously mixed. That's one way to take, that's one approach to take when we have just simple droplets, but that's not what people in labs are usually dealing with. The usual case is you have actually a protocol which can consist of, in this case, of 3 subprotocols, and that's usually expressed in natural language, human readable language, and getting that and translating it directly into actuation sequences, it's pretty tricky, especially if the protocol is large and you want to parallelize. So there is need for tools to actually get this protocol and translate it into something structured. So if we look into just the first step of this 3-step protocol, the PCR, we see that we have some inputs, we have some outputs, and then we have a description what we need to do with that. We can transform that into a structured program, a code, where we have a function which runs PCR, and again, it just goes and mimics what a technician would do in the lab when they're performing this protocol. This expression, this is capturing this protocol into a language called BioScript. And although it looks fine for people who are dealing with programming, it might be a bit cumbersome for those who are not very familiar with the programming concepts. So what we are working on is we're working on a tool which can, on a web-based application, which can simplify the process of capturing a protocol by having a simple drag and drop interface where you can just define the start of your experiment, define the fluids that you need to merge, mix, split, heat, cool, whatever, and then connect them with arrows, creating a flow chart diagram. Each step of the process can be configured with different parameters corresponding to the different operations being handled at that node. And then from here on, the tool chain can actually compile and execute the experiment on the platform. So the tool chain that we're aiming at looks kind of like this. We have on the left side, we have our protocol, which can be captured or translated either into code or a graphical representation. Then that goes into a compiler, and from there on it goes into a custom developed assembly language, which we call bioassembly, which runs on our virtual machine, which on its side can either control the digital biochip and the hardware platform itself, or just connect to a simulator and show the execution of the experiment. So the part marked in green, it's fairly well developed. The rest is still work in progress. So what we have next is a demo which actually demonstrates the capabilities of bioassembly and virtual machine and the simulator. So here you see the kind of the front end of the virtual machine where we have a bioassembly program which creates a conveyor belt where droplets are placed and are being driven under a sensor. The sensor sends their cover, and then it sends them to one of these storing locations. And that's a program which takes active feedback from this cover sensor sitting on the top of the digital biochip and monitoring the droplets. Well, that all looks good, but let's see if it works actually in practice. So the same experiment is, you'll see a video here of the same experiment, which connects the virtual machine to the real platform. And again, we have this conveyor belt with droplets. And right here, we have this cover sensor, which feeds real-time data back to the virtual machine. So the virtual machine can decide which way the droplets should go, so it can sort them based on their cover. And here you actually see in real time the readout of the cover sensor. This proves the concept of having a real-time control over the hardware platform and what value that can bring in, because in this case, it's simply sorting droplets, but we're planning in the future to expand with monitoring the droplet location through either computer vision or capacitive sensing, and provide actually data from biosensors, let's say, which run the chip, monitoring the execution of the experiments and if they are actually going as intended. So we have also tested the system with two other applications, real-life applications. The first one is space domain PCR and cell cloning, and the second one is magnetic beads ELISA. Let's have a look first at the space domain PCR. Well, I guess you all know that the PCR is a process which is used to acquire multiple copies of a segment of DNA. That's done by cycling the PCR mix through 3 different temperatures: denaturalization, annealing, and extension. And you do that 30 times and you get 1 billion copy of the same strand of one strand of DNA that you started with. We opted for space domain PCR, where we have created 3 temperature zones on our chip, corresponding to the 3 temperature regions, 3 temperatures required for the PCR to work. And the plan was just to move the droplet back and forth between these zones for 30 cycles. We did that in a closed system where the droplet was actually sandwiched between the top and the bottom plate, and the rest of the system was filled in with oil, so aiming to reduce evaporation. To do that, we had to actually extend the system, where the modularity of the system came really handy. So we added the heaters controller, which interface with the built-in heaters, and we also had to build this temperature sensor interface, which can sample in real time the temperature inside of the chip, so we can calibrate the temperature profile of the heating zones to provide good conditions for performing PCR. Well, that sounded simple, but we had many failed attempts performing PCR on that. After some chasing of potential toxicity and surface absorption, we finally managed to adjust some of the reagents, and we got the PCR working on it, although we had also quite a lot of problems with evaporation of the sample. Quickly after, we also tried to clone and transform GFP into E. coli, where we actually executed the user and transformation step on the chip. That was not that easy because some of the reagents were very difficult to move on the digital microfluidics. So with some manual help, we managed to actually get the user and transformation working on the chip. The efficiency was low, and we're still working on optimizing the protocols actually to work on the scale of the digital microfluidics. So the second experiment that we performed was magnetic beads ELISA, and that's a diagnostic assay which targets to find the presence of a specific antigen. It's a multi-step process, and it all starts with having a magnetic bead with capture antibody on that. And if you add your sample on which you're looking whether the antigen is present or not, and if the antigen is there, it will bind to the capture antibody. After more steps, it's a multi-step process. In the following steps, you build actually a complex of 2 more antibodies, for instance, and a detection enzyme on the top, which if added with if mixed with a substrate, it will generate a signal in the form of cover change. So the detection mechanism here is that if the antigen is present, you build the whole complex, all the complex of these antibodies on the top, but if the antigen is not there, that part won't be there. And when you add the substrate, you won't see the cover change. So I want to show you a video of some of the washing steps and then the cover development process of this experiment. What you saw is a droplet containing washing buffer was extracted from this reservoir and driven to the magnetic beads, to the functionalized magnetic beads, which are this red dot down there. The washing buffer was mixed with the magnetic beads and is just moved around to do the washing step. And after a few seconds, the droplet is actually extracted by just pulling it away from magnetic beads. There is a magnet sitting just in this area beneath the beads, containing them, immobilizing them and containing them into this region. Next, another washing droplet is extracted from the reservoir, being mixed with the magnetic beads, and the process repeated several times throughout the experiment, actually many times throughout the experiment. I'll jump to minute 3 now, where you see how we add the substrate to the droplet and we observe the cover development reaction. So the substrate was loaded off camera, and that's actually this transparent droplet. I hope you can see it there, being moved towards the magnetic beads. Now they got mixed, and once they got mixed, the cover development reaction gets triggered. So the next step is just waiting for the enzyme to react with the substrate and result in cover change. You can already see that the cover starts getting denser, and in a few seconds, the magnet, which is sitting beneath and keeping the magnetic beads together, will be removed. So the magnetic beads will disperse into the droplet, and it will allow the reaction actually to happen faster. Let me again skip roughly a minute forward, so you'll be able actually to see the change in cover. So that was probably 30 seconds. You see that the droplet is turning from green to yellow, which is an indicator that the reaction has happened. Good. So final remarks. We still need to work on fully developing the software stack. There's a significant amount of work that needs to go into that in order to make it user-friendly and robust and so on. We are also currently working on integrating sensors and actuators with the digital biochips, refining and developing the applications and the protocols, because just taking a standard benchtop protocol and trying to execute the same on the digital microfluidics is not always the easiest thing to do. And of course, we are also working on advancing the hardware capabilities of our platform. And for now, we are mainly focusing on the cell cloning, where when we get a single cell, we're trying to perfect the PCR where we can multiply certain genes of interest, clone them then into a vector, and then put them into a cell, getting a reprogrammed cell. That cell can produce stuff for us or can respond to different chemicals being fed up to it. So we are also working on including that into the feedback loop. So the cell being monitored of how it develops and how it reacts to different stimulus, and if needed, go all the way back to the design phase and rerun the design build test cycle. Thank you for your attention. I'm here to take some questions.
Thank you so much, Georgi. I hope I pronounced your name correctly.
That was perfect, actually.
There were a lot of questions actually in the chat. So I'm very curious about this. My background is also a little bit of microfluidics. So I'm curious to see how you solve some of these complex problems that I think everyone is dealing with in the microfluidics field. So a common question is about fluids crossing the same path. Did you see problems with that during your PCR, or is this something that your software helps you with to make sure that certain fluids don't cross the paths of other fluids? Or have you seen any contamination?
We do know that contamination happens, and there are solutions like adding surfactants and modifying the surface and the fluids itself to reduce it or to mitigate it, but we have not worked with that much. However, the software the software can actually plan ahead the droplet paths and figure out which parts of the chip are contaminated and contaminated with what, so you can actually plan and schedule your operations into different regions of the chip. We're not there yet, but it's there's actually, our group have done a lot of theoretical work on the topic, and we'll slowly start including it into the compilation pipeline that we're developing.
Great. That was a question from Alexis Casas and Luchan. And I think Alexis has another question specifically about tuning your protocols towards your droplets, right? So certain droplets might, in order to move them, you might want to apply higher voltages, things like that. So is this something that is currently still manually tuned, or how is that performed during your protocols?
So it's adjusting the actuation voltage threshold. It's manually tuned for now.
Okay.
But considering the automation level of the system, that can easily be done later on. It's actually just a matter of writing right now, just a matter of writing a bioassembly program and just seeing what is the minimum actuation voltage when you get saturation. Very
interesting. Okay, so the other thing I noticed is that you chose for a space domain PCR, right? So you're moving droplets from one heated region to another. So one question could be why not choose to perform heating and cooling in one area specifically? And I think Lucas Boyherl is also asking how you perform the cooling. Was that done with a fan, for example, or how exactly was that performed?
Space domain PCRs looked like the faster way to do it, because we consider the droplet having a very small thermal mass, and since it moves into oil medium, it would reach its destination temperature rather fast once you bring it to the corresponding temperature area. So cooling was not performed on the droplet; it was just moving the droplet to the preheated area and just keeping it there for the predefined amount of time for each step. Space domain PCR, also we think it's kind of faster to perform in this respect, because you don't need to wait for the system to cool up, to heat up or cool down.
Nice. And then so cooling was performed with a fan or an auxiliary fan to help cooling of the system?
The auxiliary fan was just there to make sure that the system dissipates enough heat, because the electrode drivers of the system, they started heating up, and it was just skewing the temperature profile that was already on the chip.
Very nice. Okay, we have a minute left for one last question, so I'll ask the last question in the chat from Michał Galdziki. He's wondering how you can draw samples off of the chip into other platforms.
Well, you can bring them to these reservoir wells, which we haven't shown here, and just pipe them out. Or eventually the digital microfluidics can be paired with a continuous flow system, which will be able to transport the sample to another analytical instruments.
Yeah, I guess this is also one of the key challenges in a lot of microfluidics, right? Where it's how do you bring stuff to the device and how do you take stuff off, right? It's this world-to-chip interface that is quite an interesting challenge. Thank you so much, Georgi, for this wonderful talk. So we'll move on to our next speaker. It's right about time for that. So I believe this talk will be given by Joseph Wheatley. Great, you're here. He's from the University of Warwick, and your talk will be about cell-free transcription translation systems. And I believe you did whole phage assembly, and you're studying that under an electron microscope. So that's quite interesting. I'm looking forward. Go ahead.
Thank you very much. Okay, can you see my screen? Yeah. Okay, hello, I'm Joe, and I'm at the University of Warwick. I'm a PhD student there, and today I'll be presenting my work, which is titled An Investigative Platform Comprising Cell-Free Transcription Translation and Electron Microscopy for Studying Bacteriophages. So just to give a bit of an introduction of the key themes of this work. So firstly, electron microscopy, as everyone will be aware of, is a very powerful form of microscopy. And it's useful when you want to study something as small as a bacteriophage. The ones that I study are about 50 nanometers. That's the electron microscope. The cell-free transcription translation system. It consists of a crude cell extract. In my case, it's from E. coli. And then you add an energy solution and an amino acid mix and a few other additives. And it allows the transcription and translation of DNA without the cells. And in the sort of the past decade, these systems have been optimized for whole phage assembly. And this is something that I've taken advantage of for my work here, because it provides a really controllable environment for your phage assembly, where you have control over lots of different parameters that aren't available for you in vivo. And then finally, the bacteriophage that I'm studying is called K1F, and this is a bacteriophage that infects and kills E. coli K1, which is a pathogen of interest because it causes a range of different diseases in humans. So for example, neonatal meningitis and sepsis in babies. So it's a pathogen that we'd like to be able to detect and to kill in real life. So that's the sort of the wider scope of this work. So the question, why combine electron microscopy with cell-free TXDL? So TXDL lets us precisely quantify the start and end points of DNA expression. So the start point is literally when you drop in your DNA. So in my case, when I drop in my bacteriophage genome into the reaction, that's the start point of expression. And the end point is when I add antibiotic rifampicin, which forces the stop of the reaction. So that's really useful to be able to precisely quantify these time points. And then these different, and then by using this, I can obtain different time points from the TXDL reaction and visualize them on the electron microscope, which allows me to view each stage of the phage assembly. And this wouldn't be possible in vivo due to the nature of phage propagation, in vivo phage propagation, which for starters, you would need to lyse your cells, which is quite a messy process and can disrupt the state of the phage. So it's really useful to not have to do that. And finally, there's the combination of these two things. There's the potential for it to be a highly accurate tool for visually characterizing or studying phage in real time, or as close to real time as you can get, as a sort of alternative for something like live cell imaging for human cells. This could be as close as we can get for bacteriophages. So before I could start on the EM work, first I had to assemble my phages in cell-free, and for this, to begin with, I used the MyTXDL kit from Arbor Biosciences, and it needed a couple of optimizations. Firstly, the genome, and then secondly, the PEG 8000, which acts as a molecular crowder to mimic the cell environment. And once those two things have been optimized, I could get fairly high phage assembly in the reaction, which I measured with PFU, plaque forming units, in a plaque assay. And then once the reaction was optimized, I then did a time course titer analysis of the phage assembly, which is the figure on the right. So at various time points, I would forcibly stop the reaction with rifampicin, and then I would measure the PFU at those time points to produce this figure and know exactly how many phages I had at each time point throughout the reaction. And so just to go over the workflow of how I did these experiments, it began with a TXTL reaction, for which I added my bacteriophage genome. So my K1F genome was added to the reaction. Immediately after that, the phage protein expression began, and subsequently after that, the DNA packaging into the capsids, and then the self-assembly of the whole bacteriophages. And then essentially what I was doing while this, I would have several reactions going simultaneously, and I would stop each reaction at a different time interval with rifampicin, so that the reaction was completely halted. I would then take that stopped reaction and prepare it on an electron microscopy grid, and wash it and negatively stain it. And then it was ready for imaging on the electron microscope. So as I previously said, it's a way of being able to image the phage assembly process at different time points and sort of visually track what's actually going on with the phages at these different time points. So this is what the next few slides is showing you what I found. So the first time point that I took was 15 minutes. And from my previous experiment, I knew that 15 minutes had a PFU of 0, so there's no viable phages there, and the image agreed with that. There's nothing to be seen basically on all the images that I took at this time point. After 30 minutes, there was still a PFU of 0. However, I did start to find some capsids popping up, which suggested that the protein expression had begun. But as the PFU was 0, there's no viable phages in this reaction. So it suggests that the only protein expression was occurring at this time point. After 45 minutes, the PFU is 10 to the 3. So there were viable phages in the reaction. And interestingly, I started to find that there were these sort of capsid accumulation events where there was many capsids accumulating together, perhaps suggesting that there's something going on there with their assembly, whether it's perhaps a cooperative DNA packaging event. So they're all sort of accumulating around the DNA concatemers and packaging the DNA, or perhaps a cooperative self-assembly event. So they're assembling their capsids, or it may not be cooperative at all. It may just so happen that they are accumulating like this for a non-cooperative reason. After 60 minutes, the PFU had risen to 10 to the 7, and again there were more of these capsid accumulation events in this exponential phase of phage growth. After 180 minutes, interestingly, as the PFU had started to sort of plateau off, There were many capsids to be seen, but they weren't accumulating anymore, suggesting that these were mature phages, and the accumulation events that were previously occurring, they weren't to be seen at this stage. Suggesting that these were mature phages, and whatever event was occurring was no longer necessary. So the main result, there are sort of two main results that came out of this. The first is the result that I was looking for when I started these experiments, was that we've shown that electron microscopy and TXDL can be used effectively as a tool for visually studying phage assembly. This is the first known demonstration of this. And secondly, sort of a byproduct of this and a byproduct of studying my specific phage is that K1F procapsids accumulate during the exponential phase of phage synthesis, which, like I said before, perhaps there is some sort of cooperation event here to do with the phage assembly or the DNA packaging. And it's something that I'd like to investigate further. But the main outcome of this is that the EM and TXDL can be used for visually studying phages over time and characterizing that process. So what I'd like to look at further in the future is to investigate this, perhaps this cooperative event hypothesis, or learn more about what's actually going on with my phages there. And the first step that I'm doing at the moment is adding a DNA marker to the grids, so that I can visually see where the DNA, where there's sort of a dense population of DNA on the image, and whether these packaging events or assembly events are occurring around the dense DNA areas. And secondly, I'd like to use this tool to study the engineered phage that I've made in the lab, and that's the other sort of section of my PhD work is engineering K1F bacteriophages to be used as a diagnostic tool, and the way I'm doing that is by modifying their capsids. So it would be interesting to learn whether these capsid modifications have an impact on the phage assembly process. And now I have this tool where I can see if there are any visual impacts on the assembly process, and investigate that a little further. Finally, I'd like to acknowledge my supervisors and my fellow lab members. And if there are any questions, I'll be happy to answer them now. Thank you very much.
Thank you so much, Joe, for this beautiful talk. That was fascinating. I didn't know much about this topic, so maybe let's see what kind of questions there are. We can start with talking a bit more about the cell-free protein synthesis. You mentioned that you optimized the reaction, correct? And there's a question from Mahna Sabeti about whether this was optimized specifically for phage protein expression, and what were the key parameters that you optimized, and they are particularly interested in PEG concentration, if that's something you looked at.
Yeah, sure. So for this specific work, I chose to use the MyTXDL kit, which is a commercial kit made by Arbor Biosciences. But we have achieved phage synthesis from our in-house cell-free system as well. But at that time point, it wasn't powerful enough for me to use for this work. The optimizations for the in-house system, the additives that really made a difference were maltose, and then also PEG, and PEG was especially important for the MyTXDL kit as well, because if there was no additional PEG added to the kit, then the phage assembly reaction just wouldn't work at all. As you started to increase the amount of PEG, then you got sort of a vast increase of phage synthesis. So yeah, the PEG concentration was by far the most important additive for this. And I think that the previous papers that have been that have been written on phage synthesis and cell-free would also agree with that PEG is one of the most important, if not the most important additive.
Right, good thing that you could confirm that yet again. A question I had was, you mentioned that you perform the cell-free reaction and you take samples at different time points. What do you do with those samples? So you mentioned you stop the reaction, and then do you immediately do the electron microscopy, or is there any way of preserving, I guess, the capsids that are in that fluid, I guess? Like, can you freeze these, or is there any other ways of preserving it?
Yeah, you can preserve it, because once the, I used rifampicin to stop the reaction, which is a transcriptional inhibitor. So it immediately halts the TXTL reaction. So once you've added that, you can hold the reaction in the fridge at 4 degrees. I didn't try freezing it because I'm not sure whether the freeze thaw cycle would have a sort of a negative impact on the state of the bacteriophages. But yeah, you can hold the reaction in the fridge. But when I did it, I would do the cell-free reaction throughout the day, and then stop it with the rifampicin, and then go straight to the electron microscopy unit and prepare the basically take 10 microliters from my cell-free reaction and add that directly to an EM grid, and that was my workflow for it. But you can store it at 4 degrees.
Okay, interesting. Yeah, so that might be interesting to be able to preserve samples from that same aliquot to do other types of microscopy studies. And I think you mentioned that you're looking at integrating some type of marker to perform other studies, right? What do you kind of know what kind of marker you're looking at to try and integrate in the DNA?
Yeah, so I haven't fully started those experiments yet, but I have an idea of the marker that I'd like to use, and it's essentially a DNA marker that binds to a region of the phage DNA that codes for its capsid protein. And essentially, when you add that marker, the areas where the DNA is dense are shown on the electron microscope image. So yeah, it would be interesting to see where the DNA is dispersed throughout the reaction, and whether it's more dense in areas where the phage capsids are accumulating. Yeah,
amazing. Well, we're very much looking forward to seeing what more comes out of this research. So thank you much, Joe. Thank you so much. We have to move on to the next speaker. Our next speaker is Sahan Liyanagedera. They are a third-year PhD student at the School of Engineering, University of Warwick. And they're also working on cell-free TXTL systems and bacteriophage engineering, but this time more focused on therapies and diagnostics. Are you here?
I am.
Great.
Joe needs to maybe stop sharing his screen.
Yeah, sorry. Let me move that. Stop share. There you go. Sorry.
Okay, perfect.
Yeah, so the full title of your talk is Engineering SpyTag Bacteriophage K1F for Directional Immobilisation.
Yeah. Thank you very much. Can you see my screen now?
Yeah.
Okay, great. So my name is Sahan. I'm also working in the same lab as Joe in the School of Engineering at the University of Warwick. So today I'm just going to talk to you quickly about the engineering of Bacteriophage K1F for directional immobilization. So as we know, there is a massive problem with the use of antimicrobials, with antimicrobial resistant superbugs causing around 700,000 deaths per year. And this number is projected to rise over the next 30 years, and it's projected to increase more than cancer-related deaths as well. So as we know from the current global pandemic, there is a massive need for point-of-care diagnostics, and currently there is no rapid diagnostic tool available for precisely knowing what kind of bacteria has caused an infection. So if you're thinking about the end user product, we would like something like this to really be very easy to use, be able to give a result within 10 minutes, and be able to work with small sample volumes, and at the same time provide a readout on multiple different strains from the same sample. And in our group, we are basically looking at the immobilization of K1F bacteriophage. So I'm not going to go into too much detail about that, but trying to combine phage with microfluidics, because microfluidics provides the ability to have small sample volumes, increased sensitivity, speed of detection, and then through the design of the device, you can have orthogonality as well to be able to give multiple readouts from the same sample. And in our lab, we're working on, as Joe was talking about, self-reassembly of different types of phage. And while self-reassembly is a very useful tool to be able to study the assembly events, it's also got quite a good system to be able to try and package signal molecules into the capsid head, as it is an open environment. And so these three technologies are what really would allow for a point-of-care diagnostic device based on phage to be made. And one aspect is the self-reassembly. The second is to be able to create a bacteriophage which can be immobilized. So in this case, we're talking about the K1F phage and the incorporation of a SpyTag protein onto the capsid head. And then finally, how do we then get that phage to stick onto a PDMS-based microfluidic device in such a manner that the phage are directionally immobilized with their tails sticking out and into the channel, so that we have maximal efficiency of infection of the sample. So I'll first talk about the way in which we created the phage. So basically, we decided to target the minor capsid protein of the phage. So if you look at this little phage down here, the minor capsid protein is the protein that occurs at the apexes of the capsid head. It's called the minor capsid protein because it occurs less abundantly than the major capsid protein, which forms the most of the capsid head. The minor capsid protein is much larger than the major capsid protein, and the minor capsid protein is created as a translational frame shift of the major capsid protein. So you basically have a ribosomal slippage of a -1 backwards, and then that results in an additional 44 triplets being added to the end of the major capsid protein to result in the minor capsid protein. So what we decided to do was to create a donor vector, which contained our gene of interest for insertion, and then flanking it, we had a homology region for gene 10, which is the major capsid protein gene, and then gene 11 as well. When we have this vector inside the host, we infect it with the bacteriophage, and that results in homologous recombination events. And the population of phages that are produced from that infection cycle is of two types. So you have the wild type genome still present, but then you also have hopefully the integration through homologous recombination. And then we take that wild mixed population and infect it into a CRISPR selection strain. We do this about 3 times. And in the CRISPR selection strain, we have a plasmid which has all the CRISPR machinery coded in it, and also guide RNA that will target the wild type genome. So after a few rounds of selection, we're able to then isolate and identify the SpyTagged phage, so recombinant plaques, which are stable phage particles incorporating the SpyTag on the minor capsid protein. So using this method, we were able to develop 2 different phages, one which has a SpyTag, one which also has a GFP SpyTag. So that was the first part of the work, to first create a engineered phage that can be immobilized. Now, in terms of the immobilization strategy, we are obviously working with the SpyTag and SpyCatcher system, which I'll talk about quickly right now. So SpyCatcher and SpyTag are basically 2, it was initially one adhesin protein that was engineered and split into 2. And when they come together, they form a isopeptide covalent bond, which is irreversible, very strong interaction. And what we decided to do was we take the SpyCatcher and fuse it to the hydrophobic terminus of a bacterial hydrophobin called BSLA. And BSLA is a very interesting protein because it has this very highly defined regions of hydrophobicity and hydrophilicity. So when you have this protein and you have SpyCatcher fused to the N-terminus where the hydrophobic part is, if you then make a droplet of that protein, you get the proteins orienting themselves around the droplet in such a manner that you have the SpyCatcher proteins exposed onto the outer surface. And this allows us to then move on to trying to capture these droplets onto a PDMS-based device. And this is done very simply by essentially dropping a droplet of that SpyCatcher BSLA fusion protein onto a hydrophobic glass slide. This results in the orientation of the SpyCatchers outwards. And then we dry that droplet down on the glass slide. And the best way to imagine this is as if it was like a football that has been deflated. So the monolayer stays intact, but then the water has evaporated, but the directionality of the SpyCatchers are still oriented outwards towards the droplet, away from the droplet. Once that's dried, we then pour the PDMS on top. And once PDMS is poured on top, we set it and then cut it out. And we have now a chip, which in specific positions, we have SpyCatcher protein surface exposed. Okay, so this was just done for demonstration purposes just to show how the SpyCatcher BSLA protein dries to form these deflated balls. Essentially, this was a large droplet of protein, which then dries down, and what's remaining here is an empty sac, but you can see here, there is still a bubble there. And if I pop that, there will be more protein coming out of that. So this is actually used as a mechanism by bacteria to protect their biofilms from getting wet. And we're trying to use this to immobilize surface display SpyCatcher. So this is a very simple method to be able to decorate a PDMS chip with surface exposed SpyCatchers. And after we've established that method, we decided to then try and see how good it is at capturing any protein that's tagged with a SpyTag. So if you look at this image over here, in the first 3 positions, we've incubated GFP without a SpyTag, a much higher concentration onto a PDMS chip. And then there's a blank well here. And then on these ones, we've incubated GFP SpyTag at a much lower concentration. We let that incubate for about 15 minutes to 30 minutes. And then after it's incubated, we thoroughly wash it. You can wash it under the tap. You can start scrubbing it, and it will result in the retention of the GFP SpyTag. And the GFP on its own will mostly wash away, but you do see some passive absorption into the device. The good thing about this method is that really you can actually pattern the device very simply by pipetting droplets into positions onto the device before pouring the PDMS on. And in that way, you can really decide where you want any protein to be immobilized on the surface of the device. One of the important things here is to show that the BSLA protein is actually very essential to enable high capture efficiency. So this is a very simple microfluidic device. We have an imaging chamber here, input chamber, and we basically spotted SpyCatcher BSLA or SpyCatcher M-Cherry, and the SpyCatcher M-Cherry and the SpyCatcher BSLA are about the same size, about 45 kDa. And we spot them here, here, and here, let them dry, pour the PDMS on, and then peel it off. And then when we examine them, we see that despite the fact that we use a much higher concentration of the SpyCatcher M-Cherry proteins at 40 mg per mil here or 4 mg per mil here compared to the 1.5 mg per mil for SpyCatcher BSLA, the ability for the BSLA protein to orient the SpyCatchers on the outside of the PDMS means that we have a much higher capture efficiency here. So next we went on to try to immobilize our phage. So this is initially immobilizing our K1 SpyTag GFP onto the device. And then once it's immobilized, we do 2 liters of washing. Okay, so that's 2 liters of buffer into a beaker with the device, and we stir it for a bit, and then 100 milliliters at a time until the 2 liters are up, and then we sonicate it to get rid of any passively diffused in phage, like over here on the corner, you can see that. And once we've done all of that, we get rid of a lot of the passively absorbed phage, but then we still have the retention of a lot of active bacteriophages. And luckily that even after all of those washing steps, there is a massive retention of the ability of the phage to be able to kill its host. So this is some of the preliminary work that we've done. And in the coming weeks and months, we hope to do some scanning electron microscopy to show that we've actually directionally immobilized the phage onto the PDMS by getting a nice picture of it, and then try to characterize the capture efficiency between the phage and the device, and ultimately be able to build a proof of concept device for the detection of E. coli K1. And I'd just like to thank all my supervisors and collaborators and Joe for helping with the project, and I'll open for any questions. Hello.
Okay, thank you so much. That was a wonderful talk. I'm rushing to plug in my computer, which is at 5% now. Okay, saved. Yeah, how fascinating. I've never seen such a creative approach to trying to immobilize proteins to PDMS. So I think that's quite interesting. Let me see if there is some questions. Okay, there's a kind of a big picture question from Krishna Sanka. So once your assay works, what do you plan to do with it? I think I saw a picture of you placing the PDMS on a plate and performing an assay with that. But are there kind of ideas to be able to translate this into a product? Or how do you think people will be able to use this?
Yeah, so in my view, to be able to translate a phage-based diagnostic system into a point of care device, like one of the problems is to solve is the directional immobilization. Another one is really to be able to create a phage that has the ability to produce a signal molecule that can be detected without having to rely on the host replication of the signal, right? So like, there's been a lot of work done on engineering a phage to be able to, for example, put nanoluciferase onto the genome. So when the phage infects the bacteria then subsequently produces the nanoluciferase. But that would rely on bacterial expression and creating a device that will allow for bacterial expression to occur. And really what you would need to do is to try to cut that out altogether and be able to create a phage that is able to package a signal molecule in its final format that can be ejected when the bacteria are present. And then also to try and create like a non-replicating phage, because to be able to kind of get the licensing and stuff for this kind of device, you would need to make sure that the phage that you're putting onto a device is not going to proliferate. So it can still have the ability to infect and inject a molecule specifically in response to its host, but not replicate itself.
Interesting. Yeah, I see that. So there's a question on that as well. How do you think the phage can retain that ability to infect cells or lyse the cells, even after you pour PDMS over it and you let the PDMS cure, and then I guess you bind your phages to that afterwards. But yeah, I think there's a question about like how is it possible that the phage can retain that ability?
Like all the processing steps are done prior to the addition of the phage, right?
Right.
And then you could probably argue about how do you then take that device and then try to stick it onto a microscope slide, and classically that's done using like plasma bonding. But then there are methods to be able to stick devices together that don't need to use plasma bonding. But phage are very resilient, and if anyone's worked with them in the lab, they survive anything really.
Yeah, exactly. Okay, so maybe one other question is about the SpyTag. Why was there a choice of like? I think it's very interesting in using the droplets to place it and to retain their orientation. Kind of, I think that's quite fascinating. But why was there a decision to kind of pour PDMS over that afterwards? Have you thought of different methods, for example, stamping it, or can you think of ways of doing that rather than having to go through this step of like curing PDMS on top? And
yeah, I mean, there is a lot of work done on immobilization of proteins, but for us, this is like the simplest way to do it, basically. And it's just so like I said, like orthogonality is a very important fact, right? So in down the line, you would want to potentially immobilize different bacteriophage on different parts of the device. Yeah. And by being able to do that by hand is like quite simple. And this method allows you to do that very simply.
Yeah, it was quite fascinating. I'm surprised it works, and it's interesting that you're interested in, like, in studying further, like exactly how why this is working and whether the orthogonality is actually retained.
Yeah, one of the like the key things that allows the drop to be captured by the PDMS is the fact that the protein is kept in a carbonate buffer. So when the droplet evaporates, the directionality is maintained, but also there's like some level of salt crystallization occurring, and that that gives some grip to the PDMS to be able to capture and rip off the monolayer from the slide, and then
interesting.
The PDMS.
Okay, one quick follow-up. I see we're over time, and maybe I'll let people know who want to head off that in 15 minutes, so I guess 12 minutes from now, there is the next keynote by Dr. Thomas Gorochovsky about programming biology, controlling the flow of molecular machines, and powering life. I will continue with just 2 little more questions because it's a break is following anyways. So have you, what about controlling the evaporation rate? Have you thought about that, or have you maybe just seen different results in your experiments, whether you perform it on a hot summer day or in a dried environment, for example?
So I mean, the BSLA protein is quite stable. So the key with the BSLA protein is to make sure you keep it out at a concentration where it doesn't spontaneously form the monolayers. And actually, when you're pipetting it, you have to use a single pipette tip every single time. Otherwise, you get monolayers forming inside the pipette tip. In terms of I think droplet volume is probably the most important thing. So like if you have like a much large droplet, so it never completely evaporates. So the droplets I showed you on the slides, they were about 5 microliters, and even after the PDMS being put on and being peeled off, there's still some liquid that's remaining because the monolayer is so good at kind of keeping the water out or keeping the water in. Yeah.
Fascinating. Okay, just one last question. Let's see who I didn't mention yet. Mahnaz Sabeti has a question. Do you have an estimation for how long this phage surface will be active to kill bacteria?
I think right now I don't, but as I said, like they are quite stable, and ultimately a end user device would be self-enclosed, right? So right now I'm doing it in an open system because I'm working with the device, but ultimately I'm planning to actually do the immobilization of the phage by actually flowing the phage through the device once the device has already been made. So it will be enclosed, and hopefully that will provide a bit more protection.
Okay. Yeah, great. I think you got a lot of interesting questions here, and thank you so much for your talk, Sahan. This was a great session. We learned a lot, I think. And yeah, as I mentioned, the next keynote will start at 4:15 p.m. British Standard Time, which is for me in 10 minutes at 11:15 Eastern Standard Time. So we'll see you there. The link is in the chat, and have a good break. Thank you.